So let's get started. How's everyone doing? Hope you're doing good. Hope the election hasn't stressed you guys out too much, or maybe you don't care at all. But if it does, if you do care, I do hope it hasn't stressed you out too much. So today we're going to be talking about sort of our last like intercellular sort of more specific topic of the semester, thinking about how cells communicate. So it's a relatively short lecture. I don't anticipate it's going to take the full time period to do this. We're going to talk about how cells communicate with one another. So we're thinking at like a system level here, right? We're thinking about not just within a single cell, but we're thinking about cell to cell. So let's dive in. So when we're thinking about communication, we're thinking about a couple key terms here at first. We have a ligand, where it's our signaling molecule. Now a ligand is pretty broad in terms of what it can be. But our ligands can be things like hormones, they can be ions, they can be small carbon compounds, they can be any number of different things. So ligand is pretty broad as a general term. We have receptor proteins, which are proteins essentially embedded into our cellular membranes that receive said signals. And ultimately, when we're thinking about communication between cells, it's just an interaction between these two processes, right? We have a signal being sent, it's recognized by another cell, and the other cell does something about that signal. That's the sort of the gist, right? That's the flow. A liver cell says something to my other liver cell, and it causes that other liver cell to change its behavior. So that's what we're going for here. That's the sort of overall flow of this. Now, visually speaking, we can have some sort of external environment. We have our phospholipid bilayer here, and we have the intercellular here. And we can think of a couple different things, right? We could think about some environment bind to a receptor, and that receptor causes some sort of change in a signaling pathway inside of the cell, leading to some sort of cellular response. And that cellular response can be any number of things. It could be transcription, translation, changes in the physiology of the cell, it can be any number of things. Now, we can also think something like something that's very hydrophobic, which will actually pass right through the cellular membrane. So remember that our membranes are hydrophilic on the outside, hydrophobic on the inside. So things that are hydrophobic typically pass right through relatively easily. But a hydrophobic thing will pass through, bind to an internal receptor or something that's what we call intercellular, and that will do some sort of change in the overall pathway in the functioning of the cell. Now, when we're thinking about cell signaling, there are four basic mechanisms by which cells can communicate with one another. We have direct contact, we have paracrine, endocrine signaling, and then we have synaptic signaling. And there's within cell signaling. So like my liver cell sends a signal to itself, which is what we call autocrine signaling. So we're going to focus on the main four, and you probably have heard of these in some shape or form, in particular, probably this endocrine signaling idea. So let's talk direct contact. How do cells contact one another? And there's a couple different ways by which cells can contact or be connected to one another. First is what we call direct contact. By what we call gap junctions. And so these rely on direct surface to surface contact. And we have a receptor essentially on one cell that's basically connected to a receptor on the other cell. And so these are basically channels that allow passages of these messages straight through these membranes, straight from one cell to the next. These are also pretty important in early development. So a lot of your early developmental cells, as you're going from a single cell to multicellular, have tons and tons of these signaling pores to send things directly between your cells. Again, as paracrine signaling, which is where a signaling is released directly from a cell and has an effect on a neighboring cell. And so you can think of, we have some sort of cell, secretory cell here. It secretes some compound that is then stimulating something in the neighboring proximity to it. And what this is doing, when we're thinking about paracrine signaling, it's signaling things that are in tight clusters. So you're thinking about maybe a couple cell radius of a cell undergoing paracrine signaling here. Excuse me. And we typically see this type of signaling between our immune system as well. So then we have our endocrine signaling. Again, this is one that's probably you're very familiar with. You've probably heard of this in some shape or form. But our hormones, so the ones you've probably heard of are like testosterone and estrogen and stuff like that. There's lots and lots of hormones. But the endocrine signaling pathways travel through circulatory systems. So this can be your blood vessel. You could also think about like a plant. You could think about plants have circulatory systems as well. Not like blood like we do, but they have a circulatory system, very much like we do. And so we think about the endocrine system as sort of long-term signal. I create a signal in my brain, not like a chemical electrical signal, but like a hormone signal in my brain, and I can send that signal through my vascular system all the way down to my feet. So I can change my behavior in one place and then cause it to change the behavior in another place. So it's a pretty extensive mechanism. It's basically your long-term signaling. So if you're going to coordinate something between your heart and your kidneys, your feet and your head, you're all about this endocrine signaling pathway. Another pathway, again, one of our four that we're talking about that you've definitely heard of is the synaptic signaling pathway. This is something that occurs only in animals and only in higher animals that have nervous system. So this is signaling between nerve cells. So you've probably heard of like serotonin and dopamine and stuff like that. Well, those are what we call neurotransmitters. So we have a nerve cell releasing one of these neurotransmitters, again, like serotonin, dopamine, so on and so forth. They release it into the sort of the void in between the cells, like the space, and it causes a binding. So that void is what we call the synaptic gap, and these signals bind to another nervous system. Uh, neurobiology is a pretty exciting field, just as a note. So all this, if any of what I just told you sounds cool, there's lots and lots of uh, synaptic signaling that we're not going to talk about today that I would recommend that you take a peek at because it's pretty cool field. I will say. Now, those are four sort of main types, but the process we're going to be really kind of keening in on today is what we call signal transduction. And this is where we have events that are occurring in a cell in response to a signal. So what we've been talking about thus far is when a ligand binds to a receptor and that induces some sort of cellular response. Now, cell types can have conserved responses. So uh, you can have, for instance, you can have a signal from like glucose or a sugar or a hormone that could be conserved across your different cell types. So like if your liver cells encounter glucose, they behave the exact same way, they have the exact same signal response as say as your brain cells encounter glucose. However, different cell types do and more commonly respond differently to the same signal. So epinephrine, you guys have probably heard of that, it's adrenaline, right? That affects your signaling pathways inside your cells differently depending on what it is. So epinephrine affects your colon cells differently than it affects your brain cells. It can either be a conserved response or a completely different response. Now, the most common response we see inside of a cell is what we call phosphorylation. Now, phosphorylation, just breaking it apart, phos is just phosphate. Phorylation basically is just the addition of. So we have the addition of a phosphate group. So phosphate being PO4 should be something you recognized from chemistry at some point. But that's all phosphorylation is. Now we'll talk a lot more about phosphorylation in a couple classes when we talk about respiration, but it is definitely in a process for discussing how signals are essentially passed in a cell. Now, phosphorylation is a really common way to change the activity of a protein. Simply by tacking on a phosphate group, you can completely change the activity of a given protein. It's a really sort of important mechanism here. So there's a couple of enzymes that we should make mention of, and these are broad classifications of enzymes. But anytime you see a kinase, typically, the most common you'll hear about are histidine kinases, but these are enzymes that add a phosphate group to a protein. So anytime you see kinase, it's always thinking about adding a phosphate group to a protein of interest. So that's the first type. The second one is what we call a phosphatase, which is an enzyme that physically cleaves off or removes a phosphate from a protein. So those are our two sort of key enzymes that you got to keep in the back of your head. But what does this physically look like? Well, if we have just a example here, it doesn't really matter what it is, but we have a kinase, right? That's what's adding a phosphate. So we have this group here, this amalgamous protein here. We have a kinase converting ATP to ADP, so it's cleaving off phosphate group. So just to note, we've talked about ATP a little bit. ATP is adenosine triphosphate. It's the energy currency of the cell. We'll discuss it a lot more in a couple weeks. But we produce ADP, which is adenosine diphosphate. So we're going from 3 phosphates to 2 phosphates here. So this enzyme cleaves off this phosphate group and just tacks it onto our enzyme. Now, a phosphatase would do the exact opposite. It would cleave this phosphate group off, creating a free phosphate group, returning the protein back to its original state. We have this dichotomy, right? A kinase adds a phosphate group, phosphate removes one. Now, I will make mention, adding a phosphate group can do one of two things. It can either add energy, which is an important, most things require energy inside your cell, or it can cause a change in the shape of the protein. So it does one of the two things. So those are the sort of the key sort of proteins. We also have a couple other types of proteins that we need to discuss, what we call receptors. And so we have intracellular. So these are receptors or proteins that are located within the cell, so in the cytoplasm. And then we have cell surface receptors, or what we also call membrane receptors, which are located directly on the plasma membrane. And what they do is they bind to stuff that's outside of the cell. Now, most commonly when we see these receptors, they are what we call transmembrane. They're not just like embedded on one side of the membrane. They typically span the entire membrane, so they connect the inside to the outside for these types of proteins. Now, when we sort of break down some of these membrane receptors, there's a couple of classes that we need to talk about in relationship to cellular signaling. And so the first is what we call chemical gated ion channels. Try to say that 5 times fast. It's channel-linked receptors that open and let a specific ion pass in response to a ligand. So something binds, it allows like a hydrogen ion to pass or a chlorine ion to pass. That's what we're talking about. We have enzymatic receptors. So this receptor is an enzyme that is activated by the ligand binding. And most of these enzymatic receptors are what we call protein kinases. And so just remember, protein kinases add a phosphate group to another protein. Then finally, we have our G proteins, which is probably of all these proteins. It's what we call G protein coupled receptors. And these are a G protein, which is bound to GTP. Now, you might recognize GTP being related to ATP, and you would be right about that. It's not adenosine triphosphate, it's guanine triphosphate. So it's not adenine as a base, it's got guanine as a. So we'll talk about GTP more in a little bit, but some also fun knowledge from the biology world here. But what these G proteins do is they assist in transmitting a signal from one receptor to an enzyme. So we would call this vector protein as a sort of a broad class. So we could just look at this. So what we have these gated ion channels here. We have a ligand binds, it allows ions to pass through. We have a ligand binding, it turns the ligand to bind, it causes a change in the activity of something leading to a cellular response. And then we can look at our G proteins here, where we have a ligand binding, where it causes essentially a cascading reaction, where it causes a conformational change in one protein to change another protein. Uses GTP to allow something to happen. So this is our 3 classes of proteins that we typically see in terms of receptors. Now, those are what are bound to the membrane. As I mentioned, we have intercellular receptors, or intracellular receptors. And so the most common ones we see are what we call steroid hormones. And so these are common, the most common intercellular receptor we see are in response to steroids. So steroids are just they're just pretty common. They're non-polar, they're lipid soluble, they're carbon compounds. So they're pretty common, right? So steroids have kind of a negative connotation, right, from like baseball and like the Olympics and stuff like that, right, as being bad things. But steroids are really important for like signaling and structural components of your cells as well. So don't think because you see steroids, it's a bad thing. In biology, there's lots of uses for steroids, not just being shady with your athletics. Now, steroids can directly cross the plasma membrane, so they enter directly into the cytoplasm. And what ends up happening is these hormones bind to the intercellular receptors, causing this complex to shift from being in the cytoplasm to the nucleus. And hormones, what they typically do, they could do a couple things, but the most common thing they do is act as a regulator of gene expression. So let's just look at a sort of conceptual diagram here. We have a hormone, we have its intercellular um, um, receptor here. So we have the hormone diffusing through the cellular membrane. It binds to its spot in its signal molecule. Um, binding to this causes the inhibitor to detach. So you might recognize the inhibitor from last week as we talked about like inhibition of transcription. Same kind of thing. It inhibits the protein from doing what it's going to do. And the only way it's inhibited is a steroid is not bound. It causes the inhibitor to fall off. And then we finally have our protein. Once the inhibitor is off, it localizes through one of the nuclear pores into the DNA so that it can thus affect transcriptional responses. So you can imagine like you have a hormone, it needs a specific protein produced. Well, hormone diffuses, binds, causes a change in transcription, leading to a cellular response. Simple sort of idea, but as I'm sure you guys know, hormones are really, really powerful, and steroids are really, really powerful sort of compounds, right? They do a lot. Hormones control almost everything about you as an individual. They don't just make your voice squeaky as a teenager or make you hairy, right? They control everything. So this pathway, again, seems simple, but really important for the functioning of a large individual such as yourself. Now, we'll dive a little bit deeper into what a steroid hormone receptor looks like. It has three functional domains. So it has the hormone binding domain. You can see these here, right? One, two, three. It's got three components. It has the DNA binding domain, and then we have the domain that directly interacts with coactivators to affect the level of gene transcription. Now, when these hormone receptors are in an active state, the receptor itself physically cannot bind to DNA because their inhibitor protein occupies that DNA site. So you can see that here, right? It physically bind because we have this inhibitor here that's, in this case, it's released, but here it's just bound, right? And you can see that it's this DNA binding site. If it had that inhibitor, it physically couldn't bind to DNA, right? It wouldn't be able to read the grooves on DNA, right? But the binding of the ligand, as you can see here, causes that inhibitor to fall off, which frees up that DNA binding site to then bind to DNA and affect transcriptional levels. So this is going to be something that we refer to as like an enhancer, right? But something that upregulate and make transcription of something faster. Now, in addition to this, we've also mentioned this idea of a co-activator, and co-activators sort of are a variable class of what they do, but the target cell of any given hormone can the hormone can affect the cell's biology in a number of different ways, depending on what type of cell it is. So, when you produce testosterone or estrogen, it affects your liver cells differently than it affects your pancreas cells or your kidney cells, right? And the interesting thing is that sometimes, depending on where you are in your liver, the hormone can affect your liver cells differently. And what causes it to affect your liver cells differently or affect your kidney cells differently than from your liver cells is these idea co-activators. And what different co-activators cause different responses to the same exact signal in different cells or similar lines of cells. So the one we're going to discuss is estrogen. It has several different effects in uterine tissue and versus mammary tissue, so breast tissue and downstairs tissue. Um, and the regulation of this is not the presence or the absence of a receptor, but it's the presence or absence of a co-activator. So we'll get there in just actually I put that slide for, but just to know, co-activators in this case are dictating how estrogen affects the signaling within inside a cell. Um, in addition, we can have not just things that affect transcription, we can have intracellular receptors impacting um, uh, sorry, just completely lost my train of thought there. Intercellular receptors don't just have to affect transcription, they can act directly as enzymes. So for instance, nitric oxide, which is a small gas molecule. It can easily diffuse in and out of cells. Nitric oxide's toxic as a note. Um, but it's a really common thing. We have lots of ways to deal with nitric oxide. But nitric oxide binds to an enzyme called guanyl cyclase. Did I say that fast? What this does when nitric oxide binds to the cyclase, it enables it to catalyze the synthesis of cyclic guanosine monophosphate, or cGMP. You can try saying that 5 times fast. And kind of cool is what cGMP does is an intracellular messenger that relaxes smooth muscle cells. For those of you that like to exercise, when you're exercising, what allows them to relax and extend and relax and extend is this actual pathway, which I think is pretty great. You're seeing an actual thing that actually lets you do what you do every day, right? Extend and contract your muscles. So I think it's pretty cool. You didn't know it, but your intercellular receptors were doing work for you all the time. Now, our other sort of key enzyme that we're going to talk about today is what we call a receptor kinase. Now, its name, as it implies, is the kinase. So they phosphorylate things, right? And when you phosphorylate a protein, it alters the function of the protein. Right? I told you it either changes the shape of the protein or it gives the protein the energy it needs to do what it needs to do. Now, the one we're going to discuss is receptor tyrosine kinase, or the RTK. When you come take cell biology with me in the future, we'll dive pretty deeply into the RTK pathway. It's a pretty important pathway for the regulation of eukaryotic cells. Also, really well studied receptor pathway as well. But you'll get a deep dive into it, and you'll hate it. And maybe next year or the year after when you take cell biology with me. But you get a shallow dive today. Just as a note, when you take cell biology with me later on, you'll notice that this course mimics pretty heavily what we do in cell biology. Just cell biology is just deeper dive into all this stuff. So, now, what the RTK pathway does is it influences cell cycling, which we're going to talk about next week. It influences cell migration, which we're not really going to talk about. And it influences cellular metabolism, which we're going to talk about in 2 weeks, I believe. And it also influences cell proliferation, which we're going to talk about next week. Now, what's interesting about RTK is that we know that mutations to RTK, because it does all this stuff with the regulation of the cell cycle and cell proliferation, we know that mutations to it can cause cancerous phenotype, so cancer. Right? RTK is a membrane receptor. And so it's not something that's just found only in animals. Plants have similar receptors as RTK. They're just labeled something different. So it's a relatively consistent thing across the domains of life. Now, RTKs, they have a couple things. So they have a single transmembrane domain, which this, as most transmembrane domains, anchors them to the membrane, which is important. Proteins flying off. They have what's called an extracellular, so something that's outside of the cell, ligand binding domain. So it's what can actually physically accept the ligand that's coming towards the cell. And then they have the intracellular kinase domain. And the intracellular kinase domain is, again, going to do what a kinase does. It's going to phosphorylate something. So what ends up happening is when the ligand binds, and I have a diagram, but I'm just going to say it first. The ligand binds, causes a dimerization and an autophosphorylation to occur, which then is followed by a cellular response, and this depends on the cellular response proteins. But let's just look at a general RTK pathway here. So we have the function, right? We have that 3-domain system. We have the receptor here for the ligand. We have the transmembrane component here, and then we have the membrane RTK proteins. Now, ligands come in and bind, and we have the overall um, um, sorry, I'm just getting tripped up on words today. They have the phosphorylation of these kinases, and what ends up happening is that these kinases then do what they do. They transfer their phosphate groups to some sort of other protein, causing that cellular response. So that's the flow. Something binds to the receptor, causes a conformational change down here in these kinases, which then causes them to phosphorylate something, thus causing whatever they phosphorylated to change the activity inside of a cell. And this can be any number of things, any number of things. And so one pathway we're going to talk about is the insulin receptor. So for those of you that I guess probably everyone knows at least one person with diabetes. So this one might hit a little bit home for some people, but insulin is an RTK receptor, and so it's basically an activated receptor that has phosphorylated sites that allow for docking. Uh, Insulin is a hormone that you guys probably know helps maintain consistent blood glucose levels. If it gets too high, it's too low, it's bad. You kind of got to keep that happy medium. Insulin is what keeps that happy medium of your blood levels, your blood glucose levels. Now, insulin itself binds to RTK. The insulin response proteins then bind to a phosphorylated receptor, and then it itself is phosphorylated, and then the signal is transmitted downstream to lower blood sugar. So we can just look at what's going on here. Simple RTK. Insulin binds, we have the receptor, we have the transmembrane domain. Once it binds, we have that phosphorylation event occurring here. In this case, we have an insulin response protein. This phosphorylated protein, you'll notice that it changes shape. It changes conformation. That's one of the things that phosphorylating a protein does. It causes it to change shape. Then it causes it to activate another enzyme. In this case, it's a glycogen synthase. And for you guys that don't remember back from our small molecules class at the beginning of the semester, glycogen is just a bunch of glucose monomers linked together. It's how eukaryotes store energy in short-term form. So long-term form for eukaryotes is fats. Short-term form is glycogen. Well, actually, I shouldn't say eukaryotes. I should say animals. Animals store energy short-term as glycogen. But what happens is you have this glycogen synthase, which causes the glucose that's in your blood to be pulled in, and then this enzyme causes all these glucoses to link together, forming glycogen, thus lowering your blood cells, your blood glucose levels. So it's a kind of a fun thing. Now, those people that have diabetes, there's a couple different issues there. There can be defective glycogen synthases. They can be defective RTK pathways, or they can have just like sort of people that have diabetes like temporarily. They can have just like slight inactivations of RTK pathways. So for those of you that again, my guess is probably everyone knows at least one person with diabetes, or maybe you have it yourself. This is what's going on. This RTK pathway is kind of messed up. So for those people that have diabetes, RTK is kind of a little wonky. So next up is going to be our friend kinase cascades. Now, kinase phosphorylation. So it's a phosphorylation that leads to a cascade of different things. Now, the one we're going to discuss is what we call a mitogen activated protein, or a kinase. It's a pretty cool way. Now, what MAP kinases are, they're a really important plasmic classification of kinases. And these mitogens, as their name kind of tells you, you might recognize mito from being mitosis. So that's something we're going to talk about next week. But these mitogens stimulate cellular division, aka mitosis. Ooh, it's nice when names line up like that, right? Now, what this cascade does, it's activated by a signaling module called a phosphorylation cascade or a kinase cascade. Interchangeable there. Remember, kinase stands for phosphorylation. And there's a series of protein kinases that phosphorylate one another in relatively quick succession. And this amplifies the signal because the kinases at each step can affect multiple substrates. Now, so this is sort of the more complex version of intercellular signaling. Now, a lot of stuff inside of your cells is regulated this way. You have these cascades of phosphorylation events, but it's straightforward. You get a signal, it binds to a receptor, causes a phosphorylation. Phosphorylation causes something to go from active to inactive, right? You just have this cascade of things being turned on by phosphorylating. It's over and over again, regulating a cellular response. And so you can think about this as being a more complex form of regulation. But you could also think about it as being a pretty safe form of regulation. You might ask yourself, well, why do I think this? Why does Dr. Kern say this is kind of safe? Well, it's kind of safe because there's lots of checks and balances here, right? There's lots of checks and balances. If you have to activate multiple proteins, that means there's lots of checks on what gets turned on and what gets turned off. So it's kind of actually a pretty safe way of turning a signal on, as opposed to one protein turns on one signal. Now, you can also imagine that because there's multiple things along the way here, this might be involved in multiple processes. And you would be absolutely right about that. There's lots of overlap between kinase cascades. So sometimes this part of this cascade can activate multiple different types of cas. So MAP kinases, pretty fun, pretty important, but ultimately it's amplifying a signal, right? So we start with a signal, it binds to receptors, and it activates stuff. Those activators activate more things, and they activate more, right? And it's activating potentially multiple cellular responses, or the same cellular response a lot. So it's a really way to quickly amplify a signal. Like you can imagine, like if if I dunked you in a really hot tub, it was like really, really hot, burning hot, right? Your body's going to want to respond to that really, really quickly. Sorry, I picked the wrong example. Torture, torture was definitely the wrong example. Let's pretend you went swimming in a hydrothermal pool because that's a thing people do. You get really hot because hydro pools are very, very warm. um, and you would imagine your body's going to respond to that heat relatively quickly. And so something like a signal amplification could say, hey, here's a signal and amplify your cellular response really, really fast so that you can adapt to something very, very dangerous like high heat. So torture was the wrong example. Bathing in a hydrothermal pool, much better example. Sorry. It's been a long day. So that's our MAP kinases. That's our cascades. Um, so let's talk about some more lots of proteins today, right? It's not. I know that's not the most exciting thing, but important concepts, right? Cellular signaling is an important. Now, our next class of proteins is our scaffold proteins, and these are thought to organize the components of a kinase cascade into a single protein complex. So what this does, our scaffold proteins physically bind each individual kinase so that they are spatially organized for optimal function. Now, eukaryotes aren't exactly the most optimally functioning cells, not like bacteria are, but this is definitely a great thing for them because it organizes and keeps a function relatively contained. But the reality is this there also puts them at a disadvantage because having a scaffold protein like this can reduce amplification. Because the idea being is only one kinase can only affect one other kinase, not one kinase affecting multiple kinases. So let's look at what that looks like. We have a kinase cascade here, right? And we have the scaffold protein. You see it's just a relatively large protein. It's bound to 3 cascade kinases in the chain, and it's affecting some sort of response. Organized, but less efficient at amplifying a given. All right. Winding down on the proteins. Not done with the proteins certainly, but certainly winding down on the protein. Now we have RAS proteins, and so RASes are classes of proteins that when they're active, when they're bound to GTP, and they're inactive when they're bound to GDP. So remember, GTP is guanine triphosphate. GDP is not monetary nature at all. It's guanine diphosphate. Now, RAS as a protein is essentially flipped on like a switch because what it does is it basically exchanges GD for GTP via via class of molecules which we call GEFs or guanine nucleotide exchange factors, GEFs, by RAS hydrolyzing, i.e., converting GTP to GDP. Now, you might recall from a few slides ago when we first started looking at MAP kinase, you would have saw RAS as the very first protein in the kinase sort of cascade. I didn't bring it up because I was going to talk about it here. RAS is the sort of the first step. So let's sort of look at this. I guess not sort of look at this, but what's this? So we have our before I go on, and then once RAS is activated, I should just mention this before we go on. Once RAS is physically activated, it activates the first kinase in the kinase cascade. Now, what we're doing is we have our inactive G protein, right? Our inactive RAS, right? And it's bound to GDP, right? Guanine diphosphate. Now, it gets turned on by exchanging GTP for GDP, making it become active, and thus RAS, or in this case, just a small G protein, and then affect multiple things down the line, and then it could be turned off by essentially cleaving off a phosphate group here. Now, the single largest class of proteins that's really important for cellular signaling is all the G protein coupled receptors. Sorry if this class isn't the most exciting. I know it's a lot of proteins, but it's just the way cellular signaling is. Proteins interacting with proteins causing things. So I apologize for the lack of exciting stuff here, but it kind of just is the way it is. Now, G protein coupled receptors act by coupling with a G protein. So, in this previous slide, you saw a G. So, and RAS is just a type of G protein. But G proteins provide a link between the receptor that receives the signal and the effector protein that produces the cellular response. All G proteins are active when GTP is bound and inactive when bound to GD. And the effector proteins by which G proteins act upon are typically enzymes. So let's look at a G protein coupled receptor function. Exciting stuff. So we got our protein, transmembrane, we have ligand, binds to the receptor. That's sort of the flow we've been going for, right? We have our inactive G protein here. And now this G protein is bound to GDP, thus it's inactive. But you can see it has 3 domains: beta, gamma, alpha, ABC. Now, once this binds, we have a physical exchange of GTP, cleaving off the GDP, thus activating the G protein, because again, it's bound to GDP. Now, what this does is it essentially casts off beta and gamma, thus associating the alpha subunit with the effector protein, which is then going to coordinate something with that cellular response. Now, you can reverse that cycle by just simply cleaving off a phosphate group here, reassociating beta and gamma, thus creating ourselves an inactive G protein because we have GDP. The reality of the exchange here, it's not, when we go from GTP to GDP, it's an actual physical replacement. They don't like add on a phosphate group, it actually just replaces it completely, which I always thought was kind of stupid. But when we make GDP, or when we make GTP into GDP, we actually remove a phosphate group, which I never understood why. That's just the way the cell does it. Not everything makes sense, but it does. Now, we have another class to talk about, which is secondary messengers, and these are effector proteins typically activated by G proteins and producing some sort of secondary messenger. So the 2 common ones that we see are I can never say this one, adenyl cyclase, there we go, and phospholipase C. Now, adenyl cyclase produces cAMP, or cyclic AMP. Now, you should recognize cyclic AMP from last class when we talked about the lac operon, right? When we talked about the lac operon, there was an association between the expression of that gene cluster and the presence of cyclic AMP. And there was lots of cyclic AMP, the expression was enhanced, right? Now, cyclic AMP is found in eukaryotes as well, and it's just essentially a part of this sort of signaling cascades that we've been discussing. This enzyme, adenyl cyclase, produces cyclic AMP. Cyclic AMP then binds and activates a protein kinase A, pKa, as its name implies, pKa as phosphate groups to specific proteins. That's all kinases do, right? Now, phospholipase C, we have a compound called PIP2. It's acted on by an effector protein, phospholipase C, which cleaves PIP2 into IP3 plus EG, which I think are some pretty sweet acronyms for compounds. but both DAG and IP3 are actual secondary messengers. So let's just look at what we have here. So the way adenyl cyclase, so making cyclic AMP, is we start off with ATP and we convert using the enzyme adenyl cyclase, CAMP, plus a couple phosphate groups. So we produce a couple free phosphate groups here. But cyclic AMP, not too different, right? We have the amino group here, we have the carbon, we have this sort of weird branch, right? Cyclic AMP as an adenosine monophosphate. Things are nicely labeled in this case. So this is what adenyl cyclase looks like. This is what cyclic AMP physically looks like. Now, DAG and the activity of phospholipase C, we start with PIP2, and this is what PIP2 looks like. And so PIP2 is kind of an interesting compound. You'll see it's got the five hexagonal sugar here, phosphate groups, 3 of them, and then it's got this sort of carbon-oxygen group bound to the plasma membrane. What ends up happening is that phospholipase C cleaves basically this in half, producing IP3, which is this hexagonal ring plus 3 phosphate groups, and then DAG is just basically this membrane-bound, sort of hunk of oxygens and carbons. But we can sort of just look at what's going on here, right? So this is cyclic AMP signaling. We have a ligand binding. We have a G receptor protein complex here. It activates a G protein. So we have the GTPs being added. You lose the beta and gamma subunits. The G protein then binds adenyl cyclase. Now, as you remember, adenyl cyclase takes ATP, converts it into cyclic AMP, and then cyclic AMP activates pKa, which then in turn activates other proteins, which thus coordinates an overall cellular response. I hope you're sort of getting the flow of things. Something activates something, it requires something to change forms, and then it activates the cellular response. That's basically how all this stuff works, right? You can imagine some original signaling pathway here in an evolutionary context, and it just evolved slightly different mechanisms over time because the mechanisms by which this occur and the structures are really similar. So there is evolutionary conservation here. So now we'll talk about sort of our next part. So we're thinking about DAG and PIP again. So we're actually going to talk about this in relationship to calcium. Now, calcium is a pretty commonly used secondary messenger. And so typically speaking, inside your cells, calcium is typically low. However, outside of your cell, calcium is typically pretty high. Now, as you should recall, calcium does not readily diffuse through the membrane. It's charged things don't pass through your plasma membrane pretty much at all without help. What ends up happening, we have our friend IP3. It will bind to receptors in the endoplasmic reticulum, which signals the release of calcium. And now calcium initiates cellular responses by binding to cytoplasmic protein, which we call calmodulin. I think that's how you say that. And calmodulin, I think that's how calmodulin binds to a wide range of proteins, like kinases and ion channels and receptor proteins. But instead of me just telling you, let me just show you. So you have some sort of ligand binds. We have the G protein, right? The G protein gives off its beta and gamma subunits to acquire GTP. GTP binds to phospholipase C. Now remember, phospholipase C cleaves PIP2 into DAG, which stays at the membrane, and IP3. Now, once IP3 is produced, it heads on over to our endoplasmic reticulum, which causes shuttling of calcium ions from the endoplasmic reticulum into the cytoplasm, which then bind to some sort of binding protein, which then elicits some sort of cellular response. So again, nothing crazy, right? I mean, this is building slightly more complex than what we started with, right? What we started with was like ligand, receptor, action. Now we're getting ligand, receptor, intermediate, enzyme, breaking down something. Right? So it's building on the same stuff. The big things here to remember are like sort of the generalities here, right? Um, now, secondary receptors different receptors, I should say, can produce the same secondary messengers. And so, for instance, hormones, glucagon, and epinephrine can both stimulate liver cells to mobilize glucose. They're different signals, but the same effect, and they both actually act by the same transduction pathway. So, um, and we can look at this. Epinephrine binds GTP, right? Dephosphorylation. We can see glucagon, same process, right? Slightly different, you know, enzymes here. Um, Slightly different enzymes here, but same process, right? We have adenyl cyclase, it's producing cyclic AMP, which leads to a cascade and a change. In this case, we're we're essentially cleaving glycogen to make glucose, and so we're changing the behavior, right? We're breaking down the storage way for storing sugar into the actual usage form of sugar. And again, same pathway, different different ligands here. So, um, so as we sort of approach the end of all this fun receptor stuff, we should make mention that there are different receptor subtypes. Now, a single signaling molecule like epinephrine can have multiple different effects in different cell types. So we've already talked about that a little bit. But what this implies is that there's multiple forms of the same receptor. So these we would call different subtypes or different isoforms. Isoform just refers to a slightly different shape. Remember, protein shape matters a lot. Changing a protein shape slightly, change in function. So we can think about epinephrine. Epinephrine, pretty important signaling hormone. It has 9 isoforms. What do I mean by isoforms again? Slight different shapes. Same chemical composition, slight change in shape. Now, it's encoded by different genes, and the sequences, when you look at the DNA sequences, right, A's and T's and G's, they're similar, but what the part of the DNA that makes the protein on the cytoplasmic side is very, very different. And these different isoforms of these epinephrine receptors activate different proteins, leading to different signal transduction pathways, i.e., different signaling pathways. So I can have epinephrine bind in one cell and say cause a breakdown of glycogen. But I can have it bind in another cell, which causes synthesis of fatty acids or something like that. So same molecule, slightly different receptor, very different response. And as a sort of our last note here, I just want to note, well, actually, this one's not all that important. So that's going to be the end of that's going to be the end of our talk on protein. I'm sure you guys are all tired of talking about proteins. I'm also tired of talking about proteins and signaling pathways as well. So that's going to be the end of our little chat on our friends' proteins. Now, let me just stop.